Welcome everybody to our 2025 webinar, or more precisely, our first 2025 webinar. Even though we announced this as this is going to be the major roadmap for 2025, and there will be a couple of roadmap things in this webinar, we have decided that we're going to do multiple of these webinars to kind of be able to dig deep into more detailed developments on the technological side, on the member side, and keep everybody up to date in a more regular fashion than as opposed to just doing this once a year. So today I'll run you through what we're currently working on, what we have been working on recently, and of course, as always, I'm more than happy to answer any question in the end. To start giving a quick overview with the next slide, so my team would need to change the slides exactly, right? And now the next slide on the other screen. To kind of give a quick overview of who we are, I'm Emil. I'm the founder, or one of the founders at Tomorrow.bio, and also a board member at the European Biostasis Foundation. So these different organizations here make up the, in quotation marks, Tomorrow Group, even though it's not officially called the Tomorrow Group. So Tomorrow.bio, as all of you know, or at least most of you hopefully know, does all the day-to-day work, right? It does the contracts, it does the life insurance partnerships, it does the cryopreservation and cryoprotection, and it does some medical development and engineering. Then the European Biostasis Foundation runs the research institute in Switzerland that does the long-term storage for cryopreservation. The Patient Care Foundation does the asset management and making sure that, the patients, if push comes to shove, if there's any problem for whatever reason in the long-term future, that there's one organization that then would be able to move those patients to another facility or to wherever. And last but not least, there's Cryo.bio, an organization that just funds basic science in the space of cryopreservation. So this is kind of the organizations that you deal with, even though, of course, from a day-to-day perspective, you only deal with Tomorrow.bio. The rest is just there to support and make sure that long-term storage is safe. As I said, questions are very important, right? So this is supposed to be a community, a webinar. So the best way to ask questions is to scan this code and then ask your question. Or you see other questions, you can upvote them if if this is something that you would like to have answered as well. 2 quick heads ups. 1, please ask the question in English. And as much as possible, please ask questions about this webinar. About general topics, or how we're set up and stuff that is of general interest, we're always more than welcome to answer those per email. But of course, if time permitting, I'm going to answer these questions as well. To dive right into, many times I've said in the past that the logistics of cryopreservation are pretty much the most important thing. Storage is great. And of course, if journalists want to reach out to us or do reach out to us, they always want to see the storage facility. But the real deciding factor is not happening in the cryopreservation facility. It's happening in the field, where the SST is happening, where the standby stabilization and transport is happening, where in our case, the field cryoprotection is happening, and so on. So 2025, just as 2024, will very much be a year where we focus on building and expanding these local capacities and local capabilities. Here you kind of see a brief overview of how we're set up. So as most of you know, of course, we started in Europe and now have expanded in the US, even though whole US coverage is not yet fully there, but stay tuned in the next update. That will probably be a topic. So right now we have these 6 locations with ambulances, 3 ambulances in Europe and a 4th one coming up. And then currently 3 locations where we partner with RBS, as most of you know, that each have a cryopreservation ambulance as well, outfitted for whole body field cryoprotection. All of them, in fact, are to be able to do the whole procedure on site to increase or improve quality. And there will be at least 1 more ambulance coming for the US and a potential other one in Europe. Now, kind of, just to show that kind of coverage expansion one more time. Europe, 3 ambulances right now, a 4th one already in construction, and the one with the green star. We currently have the plan, or at least we're thinking about having the plan, of positioning pre-built ambulances into multiple countries. So where we might not even have a team, but where the ambulance would be located so that a team from us, from Zurich or from Switzerland, from Zurich or from Berlin, could, if need be, fly in and then take over this ambulance, where then we would have significantly faster coverage with good infrastructure. Of course, the current model would be if someone dies, for example, like without prior notice in Portugal or in Madrid or in Italy or whatever, we would fly in and bring a flight kit. But these flight kits are not ideal. You can do field cryoprotection cases with them, but ambulances, our teams are more trained on these ambulances. Everything is in place, everything is a bit more comfortable, and so on. So we're looking into establishing this model where we have pre-located ambulances in other countries that are registered in that country, and then our teams would be able to take them over. So in 2025, that's the one with the green star, meaning it's not fully figured out yet, so stay tuned for more details. But the goal is to have that as an extension of our kind of emergency dispatch model. Second topic for local coverage would be, could you go back for one more slide for a second? Second topic would be, we plan to establish local CPA storage in Europe and in the US. The reason for that is that all of that is for last minute cases, right? If we have advance notice, then we of course can always bring one of those ambulances, we drive it there, it has everything on board and so on. But in last minute cases, right, if someone dies without advance notice, then all the equipment can be taken on a plane. The one thing that is difficult to take on planes is cryoprotective agent, just because it's the amount. It's like planes don't like to take these large amounts of fluids, right? In our case, a full whole body cryopreservation or cryoprotection kit, fluid-wise, would be the washout solution, MHP2, and then the cryoprotective agent. And those fluids together is somewhere in the 180 liter range. Very difficult to take on planes. So the goal here would be that in multiple locations, we pre-position that cryoprotective agent, similar to how I just mentioned of pre-positioning these ambulances, so that it can be picked up if need be. In the US, we're already doing that, and more locations are going to be coming up. And in Europe, we plan to do that as well. And last but not least, we have planned to have 1 or 2 local teams, you saw it on the slide before, 1 or 2 local teams trained. For these ambulances, I said that in the past, and just to give you a bit of an update here. So all of the ambulances that we have in Europe, they're standardized. So we now have a fully standardized setup where everything in an ambulance is the same type of equipment. It is as much as possible at the same location in the ambulance, so that teams that train with us in Berlin would be able to take over any ambulance, no matter where it is, and feel right at home. Of course, these cryopreservation cases, they're very stressful. So it is quite important that the environment is as familiar as possible. So kind of to give a bit of, like, if you look at this picture, on the left, you see, light locations for lights. There's camera locations. These cameras are always at the same spot. The lights are at the same location, right? The holding, the connectors for different parts, you see it on the right here as just an example, is the same. The coolers are the same. Every part is standardized between ambulances. The perfusion circuit, of course, is standardized. That helps us with 3 things. One thing, the most important one I already mentioned, the kind of familiarity of the standby team or the team that would take over. The second point is it helps us to reset ambulances. So once a case happens, of course, a lot of stuff is being used, so we can just plug it in and out and reset these ambulances very quickly. And last but not least, it helps us build new ambulances quick. In fact, with one local team, we currently are buying everything and build an ambulance in quotation marks for them, so that the local team has then a standardized ambulance as well. And once that is done, I'm pretty sure I'll share some pictures of that as well. Now, going on, the SST improvements in general is always a topic we're working on. Right? And probably this will not be the last time when I will say that this is going to be our big goal of 2026 or 7 or 8. This will always be a topic. But just to very briefly catch you up on what has recently happened, we expanded our medical team. 1 additional doctor joined us. We're bringing on another nurse. So team expansion will always happen. The more members we have, the more teams we need. Anyway, we would like to have more people to be trained. Said it in one of our prior talks. To my understanding, we still have the most in-house trained people of any organization in the world. I think we're currently the only one who has doctors, at least full-time on staff. But we're going to expand that, of course, more and more. Then we recently made, I think, a very, very important change in how we train. people. So in the past, we did trainings, of course. Every organization does trainings, and everybody should. But we have moved from trainings to fully simulated cases. Simulated cases means really from A to Z. And so including driving, including random locations where the team needs to drive to. We still do trainings on new equipment, but every other training has moved into a simulated case. The reason for that is that cryopreservation cases are, and I need to remind everybody every time we talk, are kind of emergency medicine, or under emergency medicine conditions, right? You're in the field, stuff can go wrong, right? You might not have support from the family or from the hospital. Stuff happens. So it's very important for us that the team is ready to deal with whatever happens, both from a surgical perspective, from an SST perspective, from a logistics perspective, and so on. So if you don't do simulated cases, that kind of, you don't train that much. So we only do simulated cases from now on. We've started to have other groups join us for these simulated cases so that they also can train with us. And if anybody wants to, feel free to reach out. Then every case gets very comprehensive case reviews. I'll show that in a second. All of our trainings are recorded by 4K cameras plus additional people with handheld cameras, and then every time we do a full case review after. We have very extensive new training dummies. I'll show them probably in our next update. They're almost done. We've used them in 2 trainings so far, or simulated cases more precisely. They work very well. It's basically 3 training dummies that cover different parts of a standard cryopreservation case. One is kind of a logistics dummy, very heavy, around 150 kilos, which is in the ballpark of 150 pounds. Very annoying to move around. Again, logistics is very important in this case. Another one is an SST dummy, a dummy for chest compressions, for cooling masks. It simulates end-tidal CO2, so you can intubation and all of that is something that you can do. You can do bone injection. It has like real, animal bones in it, so that you can do real bone injection as much as possible, at least. And then last but not least is a surgical dummy that has a simulated sternum and simulated skin on the chest that you can use a scalpel on, and then the saw, and then we have pig hearts inside that dummy so that you can do real cannulation. So as much as humanly possible, these training dummies simulate real cases. Of course, this never replaces real cases and having people train on real cases, or at least on body donation cases, but at least it gets you significantly closer than we were ever before. Then we have set up a system now where we have remote case support. So all of our ambulances are now outfitted with these cameras. That has been a while, but now these cameras have remote lock-in capabilities, so that if people remotely want to support a case, for whatever reason, right? Something very weird happens that someone else might have seen, but the team on site, for whatever reason, has not seen it yet. Then we can bring external people in. We have done that once on a real case, not because it was absolutely necessary, maybe, but because it was helpful, and it worked perfectly. And then the last point, I think, is a duplicate from the training point. Right, going on. This is kind of showing, just to give you kind of a brief visual impression of these simulated cases. As you can already see, they're fully real. On the next picture, you see that we run through the whole fluids, right? Like simulated cases don't mean like we just simulate the cryopreservation or the cryoprotection procedure, the perfusion a bit. No, we run through 150 liters, right? Through the whole set of course, it's not real cryoprotective agent, but simulated cryoprotective agent to test the length of pump, to test just everything. And last but not least, cool. So there should have been a training dummy, but next time I'll show you that training dummy. Then this is just briefly showing, so this is a picture of one of these remote lock-in cameras, right? On the bottom right in this picture, you see the recording. So there's 4 cameras recording different areas of the ambulance. And then on the big picture is the one that shows this is how it would look. Last but not least, let me tell me one slide before, please. So one thing we're currently testing is a new approach to how we do cases fundamentally. When I say fundamentally, I mean the cool down part and the perfusion part. The reason why we do this is in improving the outcome of cryopreservation cases. Last time I said that initially we thought we're going to need way more time to improve the CT scan metrics. You know, for those of you who have been with us when we did our report on the beginning of the year, when we did the report of the 2024 cases, we said initially we thought that we're going to need way more time to improve how good CT scans look. And then it turned out that most of the CT scans in 2023 and then all of them in 2024 just are good, both from a quantitative and qualitative metric. So qualitative, that regard means how they look, and then quantitative, we always do these kind of histograms of how much cryoprotective agent, how much density we have on every pixel. So for that, we now need to move on for new quality metrics. And as you know, we're planning to now start to do ultrastructure preservation checks with electron microscopy. From a perspective of how the procedure is being done, the standard procedure in our case is what is called whole body field cryoprotection, right? You do CPS, cardiopulmonary support, you give oxygen, you do chest compressions, you cool down initially as quick as possible, and then somewhere around 20 degrees, right, you stop the cardiopulmonary support, and then at least not everything, but at least you take the chest compression off, you do median stenotomy, and then you cannulate the ascending aorta, and then you circulate cryoprotective agent. Now, you can, of course, improve that procedure. You can be faster, you can add more cooling methodologies, and of course, some of them we have already added, like nasopharyngeal lavage, and we also tested colonic lavage and peritoneal lavage, right? So you can optimize here and there a bit, but by and large, these optimizations are probably, at least the ones that we've done, they're probably in the couple of % range. Maybe something, or in the combination, it might be more. There's a couple of new things that are potentially coming up, like liquid ventilation and these types of things that might increase the cooling rates. But once every quarter or so, every half year, we try to think about, okay, if we rethink this topic from scratch, how would we improve this procedure in a more fundamental manner? And this is the one thing that you see here on the slide. So what we're currently contemplating to test, and we would need to test it significantly more in body donors before we would implement it into real cases, but at least just to give you a heads up what we're currently thinking about. So while CPS, on the right side it says here, first you start with CPS. Now, during the initial phases of CPS, every team member of a field team is, it's a stressful situation. They have their hands full, right? White water needs to go in, the thermocouples and the other measuring devices need to be attached, right? Chest compressions need to go on, medication needs to go in, and so on. But then, once that initial phase is done, there is time. Like now the teams are basically just monitoring and making sure that nothing goes wrong. Now, from that point, where you kind of have a bit of time until you usually start surgery, there's a good amount of time, right? Because you want to wait down until you cool a bit before you take the chest compression device off. So what we're currently contemplating is that during that time, we would initially start perfusion already with femoral cannulation. The trick with femoral cannulation here would be that you could, in theory at least, have the chest compression device and everything still running while you already start perfusing. And with that methodology, you would be able to cool down significantly faster because you already have internal cooling, right, with femoral cannulation. And then once you've cooled down, you would still add the aortic cannulation, the ascending aortic cannulation, for for you know, just better reaching the brain, because you know the aorta is just way closer to the brain, or you even have 2 cannulation points. We have some data that the rest of the body, so we focus on the brain, right? So in our cases, the brain is better perfused than the rest of the body. Of course, that's how we want it, and that's how we would optimize or continue to optimize, at least if we need to choose either or. But of course, it would be nice if the rest of the body, if we would also increase the you know, perfusion quality there. So femoral cannulation might help with that. Plus, it would very likely help with being just able to cool down initially faster, basically being able to start bypass faster without needing to take the chest compression device off. So this is something that we're currently contemplating. There will be way more information on that going forward. We will test it, and then we will also test it in dummies initially. We will test it in then body donors, and then we will, once this works, we will test it in the first real cases. It's technically just an add-on, so nothing technically can go super wrong. It might just, and this is kind of the downside, it might distract the team. But stay tuned, and we'll keep you posted on that. Then, just quick update. We have now crossed 800 members that have signed up with Tomorrow. The large majority of those are still in Europe, which of course makes sense because we've been in Europe much longer. But US is starting to pick up as well. And you know, if that growth continues, yeah, I always say it might be, I think in the beginning I said, you know, it might be 3 or 4 years down the line once we become the largest organization by member numbers. Now it's, yeah, I don't know, 1 or 2, and probably more leaning towards 1 year until we become the largest organization by member numbers. Due to us not having been around for that long, it will take more time for patients to catch up, right? Because the people who sign up are usually relatively young, and we just haven't been around for long enough to have these people, you know, grow old. But by member numbers, I think we're going to be the largest, you know, soonish. And I'll keep you posted on that as well, of course. And now, last but not least, a couple of quick updates. So this is something that has been in the making for a while. As a quick reminder, the CT scan analysis that we're doing, these are the pictures that basically come, these are the results, right? And long story short, what is green has reached enough cryoprotective agent concentration to not have ice crystals form there. What is yellow has not reached enough concentration, but there is still a good amount of cryoprotective agent. Here the parts, to always remind people every time, the parts that are inside the brain that are yellow, these are the so-called ventricles, which are fluid-filled areas in the brain, so no brain tissue, so those don't need to be green, but long story short, the whole brain is green. How this calibration is done, how you get that picture, not only that picture, but also if you do the quantitative analysis, what the CT scan gives you, it pretty much gives you just Hounsfield units. At every pixel or voxel, it gives you a number pretty much telling you how dense this point is. And now you need to relate that density data to how much cryoprotective agent is there. And how you do that is you take cryoprotective agent, or at least this is how you have done it in the past, and also how we have done it in the past and how these images are made. So you take cryoprotective agent at known concentrations, you CT scan those known concentrations at known temperatures, and then you know that, for example, 65% weight per volume of cryoprotective agent at negative 196 degrees Celsius has the number, for argument's sake, 320 or 280. And now you say 65% is what I would like to reach in the tissue. So every pixel or every voxel that has, in this case pixel, that has reached the density number of at least 280, for example, that pixel I'll color in green. Right? And then you do that for all the concentrations, and then you know what is yellow, what is cryoprotective agent of, let's say, 30% weight per volume, or 65, and so on. You actually, if you look closely on the top right of this CT scan, you see kind of like small roundish cryoprotective agents. You see one in green, one or greenish, one in yellowish, half cut off, and one in reddish. Those are those samples that we use for calibration. Now the problem, of course, is the calibration curve is made from pure cryoprotective agent, whereas what we scan is a combination of cryoprotective agent and brain tissue. So going forward, and what we're currently establishing, and I think the next CT scans will very likely be calibrated with that, it will be brain tissue that has been immersion diffused with cryoprotective agent, then cooled down, and then used that as the calibration curve. And for now, and we've done that for a while, of course, now we all CT scans, including whole body scans, will be done at negative 196 degrees, so in liquid nitrogen, because otherwise calibration is very, very difficult. Yeah, so heads up on, heads up on that. We're gonna release more data on that once we have the full curve. Very likely, we will actually recalibrate all prior CT scans, just because it is the more correct way to do. So, and then update all the case reports. And then this is how this looks, right? So this is one of the brain samples in cryoprotective agent, marked here with the red arrow that is being scanned, just to kind of give you a bit of a visual impression. And now, just two more slides, and then I'm more than happy to answer questions. Just as a heads up, it was always a topic. We already talked about this for many, many times, right? Currently, the European Biostasis Foundation, of course, is in Switzerland. Switzerland is a very safe country, both geopolitically, from every angle that you can imagine. But cryopreservation might need, I don't know, it might take 100 years, right? It might take 200 years. Who knows? Hopefully, it only takes 50, but we don't know. So, in over 50 or 100 or 200 years, right? Even a country that currently would be recognized as very safe by all metrics, some stuff might go wrong, right? There might be geopolitical problems, there might be legal issues, who knows? So the long-term goal of European Biostasis Foundation, our whole structure, is to have multiple facilities, ideally at some point all over the world, right? From Japan to, Antarctica, US, Europe, multiple in Europe, wherever, so that if something happens somewhere in the world, you can mitigate geopolitical risk and legal risk in that country. So this is the long-term goal. Of course, as I said many, many times, storage is not where you win this race, or where the big impact is being made. This is on the SST and cryoprotection, cryopreservation side. But for a variety of reasons, we have now kind of thought about it just a bit more on two angles. One would be to have backup agreements with other organizations in place. That's been a topic that I find makes absolute sense to me, so that multiple organizations come together and say, if something happens where I am, then under certain terms, and of course with approval of the board and so on, another organization would take over and make sure that the patients are safe and sound. So fingers crossed that soon we can actually announce one of those agreements. Otherwise, I'll stick with it, and hopefully at some point we're going to have one. But I think it makes total sense. And again, any organization that wants to collaborate on that, we're more than happy to. And then we've been thinking more and more about, for opportunistic reasons, if at some point we're going to have another facility somewhere. So stay tuned on that as well. And lastly, for a long time, we've been talking about the Biostasis app, the emergency notification app that connects to these Oura rings. This is a screenshot that I just took off my phone today. It's now coming up, so it's again in testing. We deprioritized it for a while just because there's other stuff was important, but at some point we want to have that fully up and running again, and developers are currently on it, and it's going to be an iOS and Android app connecting to Oura rings or other smart devices. So it's definitely coming now, and then also will be maintained going forward. So if anybody's interested, reach out to us. There's no public testing yet, but there will be at some point. And now last but not least, I'm going to remind you again, please ask any questions that you might have. And I'm going to run through all of those questions in a second. So I'll give you a couple minutes or a couple of seconds to write down the link or scan the QR code. And then I'm going to answer those questions in full screen. Yeah, and take it from there. All right, now to answer some questions. And for questions, let's go full screen so that people actually see what's up. To start, and as a quick reminder, we're going to go, can you go back to full screen to camera only, basically? You can just go back. All right, to already get started. What are the efforts to push the law further in context of cryopreservation in Germany, Europe, and USA, even that far to freeze living humans? I think this is very, very difficult. And in fact, we're not doing much in that regard. There's two reasons for that. One is that the amount of people who want to be cryopreserved is currently so small that any government body would just say, listen, why do we care about this, right? And there's potentially other, laws and regulations that might be impacted. So there's just no reason to do it. At the same time, in every country that I'm aware of, some countries are easier, some countries are tougher, but in every country there is some way of how cryopreservation can be done. So trying to get any fixed legislation or like rules or like laws for that has the risk of making something worse than it is right now. So for the time being, we're not going to get involved in that. If in the future, 100,000 people are signed up for cryopreservation, then we might reconsider. But for the time being, this is not something that we will do. And anything living humans, it's highly unlikely. So we would not get involved in this in any capacity whatsoever. So a lot of legal red tape around it. This is not possible, nor do I think this can be possible or should be possible right now. All right, next question. Will it ever be possible to store personal belongings with Tomorrow.bio to be returned to the patient upon potential future revival after cryopreservation? So this is in fact already possible now. So everybody who signs up with us, you have a small amount of personal storage at the long-term storage facility in Switzerland. Larger volumes are theoretically also possible, but would cost extra. So that is already possible today. What will happen to the business if Emil is cryopreserved? How big is the dependency on him? Will someone step up? So hopefully I'm not being cryopreserved for a long time, fingers crossed, Statistically, I'm hoping for another, 50 to maybe 60 years, we'll see. But if this happens, right, of course there's other people. So Fernando, of course, my co-founder, would take over. Then on the foundation side, there would be other people, the rest of the board, and so on and so on. So fingers crossed this doesn't happen. But since I'm well aware that this could happen, it's very, very important to me, not only for the whole organization, but also for me personally, that if something happens to me, that the whole organization stays mission-aligned, stays on course, and so on. Do you have any plans to build out SST in the UK? How would it work for anyone in an accident at the moment? Would you work with Cryonics UK volunteers? So the UK, we actually do have a good amount of members in the UK. Unfortunately, since the UK left the European Union, it's a bit more difficult to have teams there from our side locally. But it is definitely one of the locations where we would like to have at least an ambulance and then be able to fly in with a team. So definitely on the to-do list. If something happens right now in an accident, right, then we're more than happy to collaborate with any and all local organization. And Cryonics UK, of course, would be high on that priority list. Of course, we're in contact once in a while, so that would definitely be a part of our dispatch and emergency response in that case. If I'm not mistaken, there is Cryo.bio, and most of its investments is in ETH. Has switching to Bitcoin been considered? So I'm not running Cryo.bio absolutely day to day. So I think currently it has not been considered too much. I'll check in with them and keep you posted next time if this is still of interest. Coming up on the next. By the way, sometimes these questions resort. So if I'm missing your questions, then feel free to just remind me at the end and post it again. Will cryonics become a mainstream medical procedure when the first mammal, like a rat or a mouse, has been cryopreserved and brought back? Very unlikely. I'm quite usually pessimistic on how much mainstream opinion changes based on research results. Of course, a rat, being cryopreserved, being, for all intents and purposes dead and then being revived, would be something that there would at least be a lot of mainstream attention. But I don't see any good reasons yet why this would be such a big impact to the, in quotation marks, standard medical community that it would change much on for human cryonics. At least not mainstream. I think there's a chance that, maybe 10,000 people would sign up, or maybe 100,000. But mainstream in regards to millions and millions, I unfortunately don't think so. And I would be the first who would be very happy if I'm proven wrong here. How can real-time technology and first response team be promoted to ease the problem or member passing away in isolation or without support, hence bad outcome? I'm not sure. Let me read. Now the question moved. Let me check that questions again. So I think the question is about how could we make sure that if there's if someone doesn't have next of kin who contact us and so on. There's a multitude of things, right? Telling people around you, keeping us up to date if you have any medical condition or if there might be risk, like soon the app and so on and so on. So I think currently there's not this one perfect solution that you could do, but it's more a multitude of things that you can do to increase the probability that everything goes right. So for the time being, I think this is the best you can do, and then down the line, hopefully we have more stuff. Coming back from the top to recheck questions that have gotten more votes. Any news on the medicine being developed to break the blood-brain barrier during preservation to prevent information loss due to brain shrinkage? So currently, for those of you who have not thought about blood-brain barrier modification too much, currently what happens, these cryoprotective agents, they don't go easily through the blood-brain barrier. The blood-brain barrier is basically a special lining around the vessels of the brain. So cryoprotective agent doesn't easily go into the brain. It stays in the vessels. And then since it has high osmolality, it draws out the water from the brain. So that makes the brain shrink, which ideally you don't want to do. But currently, brain shrinkage is actually a sign of quality because it shows that the blood-brain barrier was still intact, meaning the procedure started relatively early and you got all the water out. So what we're currently, or we're funding research projects to do blood-brain barrier modification, meaning opening the blood-brain barrier just the right amount so that the cryoprotective agent can go out. We have gotten the first preliminary results. The answer is it's very difficult. And the reason for the difficulty is you need to open the blood-brain barrier the right amount. If you do too little, there's no effect. If you do too much, you create swelling. So stay tuned. We're keeping with that topic, so it's definitely a topic of high interest for us. But unfortunately, it doesn't seem like there's going to be easy results. Again, coming from the top to find questions with more upward. How can we participate in simulated cases? So if someone, doesn't matter where they are, Germany or any other country, if people are willing to be trained and want to be trained and then potentially would be willing to join volunteer groups or help us as volunteers or whatever, then please just reach out. Reach out to us by email, like support@Tomorrow.bio, for example, right? Or just go to our webpage and find any of the ways, right? We'll put you on a list, and then the next time we have a training for volunteers, we would invite you. Right? The only thing that we kind of, in quotation marks, require a bit, there needs to be a certain amount of dedication, right? What's unfortunately not possible is that we train all these people, right? And they just come to the training once and then they never engage with us or the topic again. So the kind of, and of course this would be kind of based on a handshake agreement kind of thing, right? We're not requiring any, legally binding commitment here, but from a handshake perspective, what we would want to have is that someone says, I'm willing to be trained, and next training, I'm coming again, right? And then if there's a case, I'll at least do my best to help. So that would be, but if that's the case, then we're more than happy to have volunteers get trained with us. So please just reach out. You're more than welcome. All right. How do you plan to introduce a more affordable option for people who are already old, 70 or 80? Maybe some loan option for younger relatives. So the fact that cryopreservation is prohibitively expensive for some people is really a pet peeve of mine. So I don't know if we don't say it that often anymore, but in the past we did a lot. So the goal of the company is to make cryopreservation better from a quality perspective, to make it more accessible, meaning more teams in more countries, and then last but definitely not least, to make it more affordable. So in our view, cryopreservation, I think everybody agrees, also the other organizations, in our view, cryopreservation should never be a topic of money. Of course, it costs money, but everybody who wants to sign up should be able to do this, no matter how wealthy or not wealthy they are. So we definitely will try to bring down the cost. Currently, if someone is relatively advanced age and is not able to fund their cryopreservation contract by any type of insurance anymore, then please just reach out. In all cases in the past where this was the case, we have found some solution, right? There is theoretically the chance to do cryopreservation cases that are more affordable. Yes, quality might go down a bit, but it is possible to do at least something. We have other organizations that we know, that we collaborate with, who brain preservation can be done under certain circumstances very affordably and so on. So if this is the case, just reach out. We're going to try to make it work. The only thing that we cannot do, there would need to be some type of proof that, from an affordability standpoint, it just wouldn't work otherwise. So it would be kind of means tested, because of course it cannot work that just someone says, I don't have the money, and then tries to get it cheaper, even though they might have the funds. So that is one requirement, but other than that, we would always try to make it work. So please reach out. Do you also work on strengthening the global regional community around Cryonics? Do you see it an important factor for achieving your long-term goal? We do to a degree. So meaning we host conferences, we host events, we of course engage with other events, we participate and sponsor other events and so on. So local teams we support a lot by trainings and happy to share equipment and so on and so on. We're happy to give talks at any local Cryonics organization in any country of the world and so on. So it is a very important part, and we do, if anybody feels like there is something that we could help with, please also reach out. How many members are in different European countries? Top of my head, I don't know the exact breakdown. The top countries are Germany, UK, France, I think. But it is on our webpage. So just go to our webpage and you find a breakdown of all. I think that there's a breakdown of the top 10 countries, maybe more. So just check our webpage. There is a plan to establish another company or some kind of trust that would be building wealth for the cryopreserved so that they have resources after thawing. Yes, there is. We've been at that for a while. It's a bit of a complex topic, not necessarily, well, first from a legal standpoint, it's not super easy, but then the second main point is that you need a relatively high amount of money initially to be able to fund that structure. So there would need to be a couple of people who say, well, I have a cryopreservation company, I'm still alive, but I'm willing to already put some money, which of course would then be your money, again, if you're being revived, or once you're being revived. So it needs an initial amount to be able to keep the structure operational, somewhere in the ballpark of 10 million probably. So if someone is interested, I think we do have a quotation marks waiting list for that. So also reach out, we'll put you on that list, and once that topic becomes reality, we would reach out. After covering the United States, which will be the next countries you will put SST? Good question, not fully decided yet. We're looking into some Asian countries, so probably somewhere in Asia would be the next. And maybe one Asian hub then would then cover multiple countries in Asia. That is currently the most likely thing that will happen. But no deadline on that yet. How can real-time technology and first response teams, we already had that question. What possibilities do you have of financing and cooperation in context of cryopreservation research and innovation projects? I'm not exactly sure what that question means. Currently, we fund research by a variety of methods, right? So Tomorrow funds research, the European Biostasis Foundation funds research, of course, Cryo.bio only funds research. We collaborate with other funding bodies. So I think these are currently the main things. If you meant something else, feel free to repost the questions and I'll come to it again. How do you collaborate with other Cryonics organizations? Alcor, CI, Southern Cryonics, Cradle, and so on. So as I said in the presentation, this has been a topic. Collaboration of other organizations has been very much something that I would like to do more of. Sometimes it's a bit difficult to get these collaborations off the ground. Again, hopefully one of these backup agreement collaborations will be ready to be announceable soon. So I think that would be the first one. Other than that, currently there's like a good amount of communication, but no like direct collaboration. Even though everybody talks to everybody, and at least we and also the others, I think, are always happy to share information and knowledge and so on. And at least we're very, very open to do that with everybody. So that's kind of the soft collaboration, like real partnerships or hard collaborations. Currently, that would be the first one. What kind of progress is possible in 2025 and in 2026 in regards to science, law, and logistics? Which one is the most important? So law is not super important. Logistics, absolutely important. Science, also very important. But science is just a slow-moving thing, unfortunately. So I think from an impact standpoint, on average cryopreservation, the logistics part would probably be the highest, right? Meaning having more teams, being faster, improving the medical procedures, which of course is somewhere between medical and logistics and science. From a scientific side, I think the biggest one would be blood-brain barrier modification. Maybe some interesting long-term things like rewarming technology and so on, but that would not directly impact cryopreservation cases right now. So I think that would be kind of the combination. But science is very difficult to predict, so we'll see. How do you plan to keep the company for 100+ years, which may be needed for technologies to develop? What will happen if the company will go bankrupt? So in our case, and that should be the case in every organization, as I always repeat myself, the operations, so the day-to-day operations that Tomorrow.bio does, is fully separated from the long-term storage. So if anything ever happens to Tomorrow.bio, nothing happens to the long-term storage. So everybody still will stay exactly where they are. The funds are totally separated. It's a non-profit foundation that is only there to make sure people stay in cryopreservation. Of course, we definitely don't want Tomorrow.bio to somehow not exist anymore or go out of business. The reason for which, of course, most importantly, is that this whole field needs to grow to be able to fund more science and build better logistics and so on. So it is an important piece of the puzzle. So we currently run the organization with the maximum amount of safety and security that you can. So from an internal perspective, right, meaning how we operate on having debt, like we have getting new supporters in, like how much years we can operate, even if no one would pay us any more money, and so on. And all of that is like years and years, right? So as much as I humanly can, I will do everything that this organization never goes out of business. Of course, in theory, this can happen, right? But as much as you can mitigate the risk, we're mitigating the risk. I think I read there was a better storage method, Doer, planned, and cost would be 250K. Is this still a plan? So the cost, we have not announced any cost, so we don't know yet what the cost will be. So there are indeed so-called ITS doors coming up, intermediate temperature storage, whole body ITS doors. Those would store at a slightly higher temperature, which in fact is better because you have less thermal stress. And hopefully one of the next updates I can show you pictures and it's already in operations. But price, we don't know yet. So price will be announced once we know, meaning we need to test it, we need to see how much liquid nitrogen consumption it has, we need to see how much the base cost of these doers is. It's definitely higher, but the exact prices we don't know yet. What are the contingencies plans for long-term storage? Given climate change, perhaps the storage facility should be moved to Lapland within the next 10 years. So the contingency plans right now would be, so Switzerland is fine for a while, right? Like even with climate change, nothing happens in the 10-year horizon. Down the line, the goal would be to have multiple storage locations, all over the world, like mitigating, legal risk, geopolitical risk, climate change risk, and so on. So that would be the mitigation, and fingers crossed, we will have some stuff to announce there soon. Have you read the recent book, The Future Loves You by Ariel? So we do a lot with Ariel. So we have written papers together. So we're in close contacts. Do you collaborate with him or with the Brain Preservation Foundation? With Ariel, yes, a lot. Again, we have published papers together. We're in half regular contact and so on. With the Brain Preservation Foundation itself, no. But with people who are involved in it, yes. I'm a healthcare medical worker in the UK. How can I get in touch to discuss any way I could work with you? First of all, that's amazing. Apart from volunteers, which also is amazing, people who are in the medical field are very, very helpful, of course. So please just reach out, right? The best way is probably literally emailing either me or support@Tomorrow.bio, or ideally even both of us. Because sometimes I get overwhelmed with emails and I'm slow to respond. So feel free to email Emil@Tomorrow.bio and support@Tomorrow.bio. And just refer to what you just mentioned, and then most likely the message will get to me, and then we're more than happy to reach out and chat and have a call. And that also holds true for all other people who want to get involved in supporting in a volunteer fashion in some capacity. In a contract, it says that storage can be refused if doing so would jeopardize the organization, like if someone famous signed up. How notorious can I be? So it's not about someone famous, if someone famous signs up, right? That would not be any problem. There is the edge case risk that in certain countries, right? Again, we don't cover these countries right now, but theoretically someone could have a case there or be citizens of that where it's just illegal, right? Where it's just not possible. And then for whatever reason, if that person would be signed up with us, right? And there is literally a risk to the whole organization that in that case it could happen. I don't imagine this will happen. It's more an edge case risk. I'm not aware of any person who is signed up with us right now where this even would be a theoretical risk. So you can be as famous as you want to be. Will you create country-specific guides on opting out of organ donation, declining autopsies, registering last will, making advance directives, and so on? Yes, we're currently working on that. So there will be a country-specific guide that gives guidance on some of these topics, even though a lot of these questions, in the end, you would need to talk to your lawyer. Right? We cannot give legal advice, of course, definitely not legal advice in some countries that where we're not based. But there would at least be some guiding principles of what you should look at. And of course, if we see it and there's some country that changes their laws and cryopreservation would not be possible anymore in that country, right? We would advise people, if something happens to you or if you get some terminal condition, maybe consider going to another country. Do you have a color-coded map of Europe for the response type, flying the team in versus driving the ambulance? We don't, but in most cases, this does not really depend on the distance. It depends on how much advance notice we have. So we had cases in Spain where we brought the ambulance, right? So we would do that even to the edges of Europe if we have time. Any plans to build SST in the Czech Republic? Currently none. But if there would be like a group of local people who want to collaborate with us, right, in an SST capacity, we're generally open to it. These agreements are sometimes a bit difficult, so it's not super easy to make them, and for us, they only work under certain circumstances, but in general, this can be possible. If you're an expert in Europe without residency, are there issues in such a case? Depends on the country. It really depends, but in theory, it would not be a problem, at least in some countries. What does the gray ambulance stand for? The gray ambulance on the. I was like, we don't have a gray ambulance, or white or yellow. You meant in the slide. So the gray ambulance means these are ambulances that are currently in construction. So they're not ready yet. They're currently in planning or already in construction. Would the foundation benefit from high-profile people being cryopreserved or talk about the plans to do so? Likely, yes. Yes, we would, right? So if you know anybody or if you are very famous, feel free to reach out. We're more than happy to check what can be done together. Why do you think that why don't you think that cryopreservation become mainstream? I do think it can become mainstream. I think it's just more difficult than... It's more difficult. The reason why I think it's more difficult is, well, cryopreservation has been around for around 50 years, right? So far, right, somewhere in the ballpark of 5,000 to 6,000 people worldwide are signed up by all organizations combined. You know, we checked, there are more super yachts in the world than there are people who have cryopreservation contracts. So it's the niche of the niche right now. So if all the advances that have been made, and don't get me wrong, a lot of advances have been made, and if the alternative is just, the alternative is cremation. It's not like the alternative is this other great treatment of aging. If given the advances and the fact that the alternative is literally cremation or burial, and given the fact that a lot of people are currently in Europe, in Western countries, atheist or agnostic, if these things together have not made cryopreservation at least kind of mainstream. And when I say kind of mainstream, I mean already, let's say 100,000 people signed up, right? If that has still, enough for cryopreservation next day, right? But bar something like that, which I don't imagine will be possible anytime soon, in the next decades even, right? Then I don't see what would be optimistic. But don't get me wrong, we're working on it. If you ask people, like, does cryopreservation make sense to you? Is this something that you want to do, right? Around 20% of population says, yes, I'm considering this, right? So the interest is tremendous. But the people who actually do this, I'm pretty sure there's a lot of people in this call who haven't signed up, right? So, and they're joining literally like a detailed cryopreservation webinar, right? So getting these people over the last threshold to actually doing this is quite tough. But we're working on it. We're trying to figure it out. We're trying to how to communicate this topic better so that more people, do or make the last step. So if you're in this call and have not signed up, do it today. Just click the button. It's very quick to sign up with us, at least the initial stages, and then we can bug you once in a while by email and remind you. But fingers crossed, we're working on it. I don't think it's super easy. Could I fund my cryopreservation by making the foundation a beneficiary of all my assets upon death? Under certain circumstances, these types of funding methodologies work. It is not possible to do a whole amount by last will, because the problem is we never know if the last will is still active, right? You could have changed it a day before dying. But with payable on death accounts and like certain trust structures and so on, it works. So in theory, it does work. Just reach out and we can talk. Do you have any plans to use new artificial intelligence developments to help with research and analysis of data, brain scans, etc.? Yes, absolutely. So, of course, not so much for research yet, but internally the company already uses it quite extensively. For research and development, it might also have, big effects. Currently, for cryobiology, it's not that great yet, or that useful. But I imagine this will get useful really, really quick, and we're more than happy. So the answer is yes. If warming up technology becomes available, how will you decide when to wake people up? So There needs to be three fundamental things before you would attempt it, right? One, it needs to be possible in the first place. When I say possible, it needs to be possible with a similar risk profile of a large-scale operation at minimum. Even though at that point, probably the cryopreservation technology will be so advanced that so if something goes wrong in the rewarming phase, right, you would just be able to put the person back into cryostasis. But of course, this is all speculative. And then you wait another 20 years until the technology has gotten better. So that needs to work. The underlying condition that led to the patient's death needs to be curable. And the person needs to have a relevant amount of remaining years of life left. So once these three things are possible, then the foundation would make the decision to attempt it. But that might be long in the future. At that point, probably there will be legal questions that need to be answered and so on. So this might take 50, 100, 200 years, like an indefinite, unknown amount of time in the future. What's the ideal focus split in percentage between technical work, cryoprotective research, legal and regal readiness, fast perfusion with minimal bureaucratic delay? Currently, it's like 95-5. Like the legal stuff is just, there is nothing really to improve, at least nothing realistically, right? Whereas on the technical side, a lot is to improve, and very realistically and very tangibly, you can work on it. So I would say, high 90s to very, like maybe it's 99% to 1 or 95 to 5, somewhere in that ballpark. Does Tomorrow.bio already cover the entire United States? No, we have not expanded to the entire United States yet. If you are in some region outside of the 3 states that we currently officially cover, which is New York, Florida, and California, please reach out to us. We just want to make sure that you understand that, if you literally die tomorrow, then we might need more time. We have, not everything is like fully 100% in place yet, but it's a matter of weeks now. So reach out. In theory, you could already sign up. How much work is needed before Cryonics becomes a real suspended animation, meaning no damage from the cryopreservation process? Bar like tremendous, paradigm shifts in research, it will take a good amount of time, like decades. Basically, so I can't predict it. It might be 50, it might be 100, but all of these numbers are pretty much the same. Like you cannot predict how much stuff will work or what will work in 50 years either. So it will be a long, long time, unfortunately. Hello, if I dead in France, what's happening? In France, we would send a team. We have like local support there. There's a volunteer team, right? So yeah, the cryopreservation would happen. Even though France has some legal complexities, but they can be worked around. What can you say about the different legal structures and regulation governing Tomorrow? Do you present challenges in expanding to new countries? So there's always a challenge, like expanding to new countries is always, there's always some legal stuff to at least look at. In most cases, this is just something to be aware of, and maybe something small needs to be adapted or something larger needs to be adapted, but fundamentally it's possible. So yeah, this is basically how we approach it. Have you done any kind of vitality test after freezing nerve cells? So on cell culture, this is being done regularly. Right? In rat experiments, it's also being done. In human cases, this is not done. Once you go to cryogenic temperatures, vitality or viability is not a relevant metric anymore. It just doesn't work well. So now, no, we don't do that. Also because the people who have been cryopreserved so far have not given consent that nerve cells are being taken. So we would only do this and exclusively do this with consent. By the way, since we're coming to the last questions, and also we are a bit over time already, if you have any last-minute questions, put them into Slido right now. Otherwise, we're going to end the webinar in a little while. And then, of course, reach out if you have any questions that we might not have covered today. Have you done any kind of, oh sorry, we answered that already. Have you tried perfusing with ethanol in the freezing medium? No, but the cryoprotective agent, it's the cryoprotective agent that we currently use would be better. If Any of you are from Poland and are interested in joining the Polish Biostasis group, please contact, email. Yeah, if there's a local group, great, absolutely join it. Also feel free to reach out. Once in a while we reach out to our members and then contact people, ask people, are you willing to be put in touch with other people from that country? We do this once in a while, so that's also a way to connect, and then if people are willing to have the information shared, then we would put all of those people in touch. All right, last chance to ask any questions. Otherwise, I would like to thank everybody for joining us tonight, and then stay tuned, reach out to us at any point, and until